With that, I'd like to go ahead and introduce Dr. Charles Rosser, who is from Cedars-Sinai at UCLA. I actually met Dr. Rosser probably back in 2017 when he was at another institution, a little sunnier, a little more tropical. So it's a pleasure to see how far Nonagen and Dr. Rosser has advanced with the assays that they're working on. His presentation is going to be a novel multiplex test to detect and predict and monitor bladder cancer. Dr. Rosser is a physician scientist and professor of surgery at Cedars-Sinai Medical Center in Los Angeles. Dr. Rosser has authored over 200 peer-reviewed publications and has garnered well over $15 million in extramural funding, largely from NCI. This was for his molecular diagnostics and experimental therapy laboratory and therapeutics laboratory. He has served as medical director of 2 large clinical trial offices, overseeing over 400 phase 1 through 4 clinical trials. In addition, Dr. Rosser has spun off 2 biotech companies, Nonagen Therapeutics, which is geared to bring to market novel therapies targeting the tumor microenvironment, and Nonagen Bioscience Corporation, which is geared to reduce the burden of cancer with innovative diagnostics. Please join me in welcoming Dr. Rosser.
All right, thanks a lot, Casey. Yeah, it's been 2017 is when we first chatted here to do our switch. So it's a pleasure to come here to talk to everyone here. I'm really interested in Steve's talk after this. I always learn so much from him, so I'm going to be very quick here with what I'm going to talk to you about. So again, a novel molecular signature to detect, predict, and monitor bladder cancer. Here's some of my disclosures. So I'm going to be wearing my hat actually from Cedars-Sinai today. You will see 1 or 2 slides from Nonagen here. All right, so my objectives will be to show you how we did this phased comprehensive approach to identify clinically relevant biomarkers and then put these clinically relevant biomarkers into a clinical grade diagnostic. So that's going to be my goals for today. So, but first let's talk about the market. If you don't have a market for your product, you'll go nowhere here. So our product is trying to address 3 things in the market. So the first thing up is BCG. BCG is really one of the first immunotherapies around. First discovered back in the 1960s that it could help cure cancer. So BCG is used to treat early stage bladder cancer, but BCG is a generic drug. And just like so many generic drugs in oncology, there's a shortage of it here. So our thought is, can we help the docs predict who's going to respond to BCG so they can get the BCG versus the ones who aren't going to respond to it? Let's pivot them to something else here. So that's one thing that we're looking at. So there's a BCG shortage here, and the BCG shortage is affecting patient care. So that's a basic fact there. Also, bladder cancer is a highly prevalent disease and one of the most expensive cancers to treat here in the United States. That's a fact too, and we'll talk a little bit more about that. And the last kind of basic tenet or fact here is that currently available urine-based diagnostics are notably unreliable but widely utilized. Let's talk about this BCG. So again, the BCG is an immunotherapy. You give it inside the bladder of a patient with low risk bladder cancer, and what will happen is you'll have this intense immune response which will then eradicate the tumor. The response rate for BCG is kind of a flip of a coin, 50-50 there. And again, I told you there's this BCG shortage. So what we want to do is we want to pivot these patients to a drug that's going to work because they have this very narrow therapeutic window. If they're not treated adequately in this short amount of time, their cancer, which usually has a 94% 5-year survival, can drop to a 50% 5-year survival. So again, what we want to do, pivot to the patient to the right treatment there. Save the BCG if you're not going to respond to a patient that will respond to it. So really it gets at the crux of precision medicine, right? Giving the right drug to the right patient at the right time. And that's what we're looking at here for our diagnostic called OnCuria. And so we're thinking an effective diagnostic to predict response to BCG would be of great benefit to the patients. So that's the first problem. The second problem is the prevalence and the incidence of bladder cancer. So over 700,000 Americans are struggling with bladder cancer at any given time. This is a prevalence of bladder cancer here in the United States. Highly prevalent disease, probably second only to colorectal cancer, that has a higher prevalence here. And then over 2 million Americans will present to their urologist with signs as though they have bladder cancer, blood in the urine. We call that hematuria. So 2 million presents with blood in the urine, and we largely evaluate all these patients with cystoscopy. Cystoscopy is a little TV camera that you can see that goes through your urethra, goes inside the bladder. Does the patient have recurrent cancer, or where is this blood coming from? Is it coming from a tumor? So it would be great if we would have a urine-based diagnostics, a non-invasive diagnostic that can help us evaluate these patients. And we do. So the CLIA labs do about 1.5 million urinary cytologies per year here to evaluate these patients. So urinary cytology is interesting. It was first reported in the literature back in 1945 at the end of the Second World War. So you're thinking, it's been going for about 70 plus years. This must be a robust test. But that's not the case here. The clinical sensitivity for urinary cytology is 35% for all comers there. So it's right 3 times out of 10, wrong 7 times out of 10. So this has caused frustrations with my colleagues, with me, a card-carrying urologist here. So this is what we want to take aim at, these 1.5 million subpar antiquated tests. We want to get that out of there and get our test into the market. So let's take a step back and look at globally bladder cancer. So bladder cancer is the 10th most common cancer here globally. The incidence of bladder cancer globally is over 500,000 people will have bladder cancer every year around the world. And the prevalence obviously even higher than that. 1.7 million people are struggling with bladder cancer at any given time. These are huge numbers. So this is showing you some of the current urine-based diagnostics that can be used to evaluate patients for bladder cancer here. And I tried to snap a line with things that are kind of singleplex versus multiplex. Here over the last several years, the multiplex has been really the push here. And this is what I'm going to obviously talk to you about. But some of the singleplex that really made a splash, bladder check, BTA stat, you can see their sensitivity numbers didn't really hold up when it was really deployed out into the clinic. So the clinical problem. Again, urinary cytology is not a robust diagnostic assay. Sensitivity 35%. It's lousy, really. Single molecular markers, the bladder check, BTA stat, have limitations, and we sort of know that. The single markers aren't going to cut, especially in something like cancer that has so many genetic defects here. So we really have to pivot towards a multiplex testing. And this was our mindset 10 years ago when we first started this in the lab here, kind of following on the footsteps of Oncotype DX at that time and MammaPrint. But now you can see everyone's really moving towards that for cancer and even non-cancer. So what we're going to talk to you today is about designing this robust assay development program, incorporating the current workflow. We don't want to ask the docs to do anything that they're not normally doing because we're not going to do it there. You're going to have a poor adoption. Then you also want to leverage the technology that's in your lab, specifically in your CLIA lab. And this is the nice thing with Luminex. So many of the CLIA labs have a Luminex-based unit there. So this is our phased development of OnCuria. And so again, it was about 10 years it took us to do this. And all in all, it's about 30 peer-reviewed publications here, going all the way from discovery all the way to clinical validation, where we'll end up landing here. So discovery. So 10 years ago, we didn't have single-cell RNA sequencing. Heck, we didn't even have RNA sequencing really at that time. We had gene expression array, and we had proteomics. And specifically, we wanted to look at the glycoproteins. Why the glycoproteins? Whether you love them or hate them, PSA, CEA, CA125, they're secreted proteins. They're glycoproteins there. So we want to enrich the secreted proteins. So my lab ended up doing a gene expression array, had a cohort of about 100 patients, 50 cancer, 50 non-cancer. And from that, we used the AFI chip. We narrowed it down to 44 genes. These 44 genes were validated in an independent cohort, also about 100 patients, 50-50 there. And those 44 genes were validated down to 14 genes via low-density array. The same time, my colleague, Steve Goodison at Mayo Clinic, was working on the glycoprotein using tandem mass spec there. Ended up coming up with a profile of 10 biomarkers there in his first group there, 100 patients, 50-50 cancer. And then that was validated in a independent cohort there, another 100, and those 10 biomarkers were validated as well. But at this time, we took a step back because we had two large data sets, the genomics data set and the proteomics data set. So we asked our computational scientists, hey, can you collate these two data sets, run your selection algorithm, and use them to see if they could kind of validate each other, right? If you had the RNA there and you had the protein there, then that's kind of a check mark that has been validated. But sometimes it didn't have to be at that same level there. So that's what he did. And so out from that came 19 biomarkers. These 19 biomarkers we then deployed in this cohort of about 125 patients, half cancer, half non-cancer. The cancers were very aggressive cancer, high grade, high stage. The controls were almost healthy controls here. And we took the 19 individual ELISA kits. So on our signature, the signature was a composite of RNA and protein. And so that's when we said, well, this is going to be a little bit difficult for the CLIA tech to analyze some of the sample for RNA and some of it for protein. So that's when we said, let's give the RNA the benefit of the doubt that it will be translated into protein. And let's look at the 19 biomarkers at the protein level in 19 individual ELISA kits. And that's what we did here in this study. The 19 ELISA kits were validated down to 10 biomarkers here. And these are the 10 biomarkers that we were left with at the end of the day, and they've stood the test of time in our subsequent studies. So these biomarkers are quite diverse, angiogenic biomarkers, inflammatory biomarkers. So you have angiogenin, ApoE, A1AT, CA9, IL-8, MMP9, MMP10, PI1, syndicin, and VEGF. So really a gamish of biomarkers there. So we're an academic lab. We were submitting papers, we were submitting grants, and we got comments back saying, okay, everything you've done looks good, but what if you have a diverse benign condition, maybe blood in the urine, but no cancer, or urinary tract infection? It's going to throw your test off. Show us that it's not. So we ended up doing this study, 300 patients, 100 cancer, 200 non-cancer. The 200 non-cancer was very diverse there. So urinary tract infections, blood in the urine, no cancer, kidney stones, something that's could wreak havoc with this test. And we set the bar for this. So again, now we're looking at 10 individual ELISA kits here. We set the bar for go, no-go at 70% sensitivity, 70% specificity. If it didn't make that, we were going to say, let's call it a day and pack it in. This one just barely squeaked by with the 74% sensitivity. Specificity was good at 90% there. So we satisfied this one reviewer there. Next, we submitted that paper, got their comments back. The comments were, this is great, but everything you've done so far has been in your own lab. Someone else should validate it. And so that's what we did. We worked with other investigators in Florida, when we were there in Florida, to help externally validate what we were doing. So they collected their own samples. We pointed them to the right ELISA kits there, and they were able to validate this. Again, about 300 patients, 79% sensitivity, 79% specificity. Submitted that to the paper, submitted it to the grant. The comment came back, this is great, but everything you've done so far has been de novo cancers, which are the new cancers. The new cancers tend to be bigger, chunkier, anywhere from 3 to 5 centimeters. They wanted us to look at the recurrent cancers. Recurrent cancers tend to be picked up earlier. They tend to be smaller, on average about 1 centimeter. So this is what we did. We had a cohort of about 125 patients, about half cancer, half non-recurrent cancer. And this shows you sensitivity 79%, specificity 88%, again, even though it's a smaller cancer. I like this one because this one actually had, we had not only cytology here results with it, but we also had the Eurovision, the fish assay by Abbott here with it. And we outperformed both cytology and the Eurovision sensitivity-wise here significantly. Again, we're at 79%, they're at about 35% sensitivity. So at this time, we had a bunch of papers published. We decided, let's do a meta-analysis, bring everything together. And the punchline from that, it's what you would anticipate, that the multiplex, bringing all 10 biomarkers together was better than any single biomarker alone, and the 10 biomarkers were better than cytology. Now, the reviewers did come back and say, hey, what if you have another genitourinary cancer, another cancer within the bladder, kidney, whole thing there? What would happen with it? So we ended up getting patients with kidney cancer and prostate cancer there. But those cancers did have individual biomarkers go up, prostate IL-8, kidney cancer, VEGF, and CA9, but nothing came back with a positive test that we were looking for, like a positive for bladder cancer. So we satisfied that one. The next one was a little bit trickier to do. They said, what if your patient has renal insufficiency or something wrong with the kidneys there? So one of the first signs that there's something wrong with the kidneys, you're going to dump a lot of protein into the urine. Well, this is a protein-based assay, and sure enough, when we had patients with renal insufficiency, it really threw off our assay there. So because that, we have now a label that's going to be on it, that if you have a GFR, glomerular filtration rate, meaning that your kidneys aren't working that well, below 45, you should not use our test here because you're going to dump a bunch of non-specific proteins that will interfere with our test. So at this time, we've done everything with individual ELISA kits. Obviously, we're not going to be able to take that to a CLIA lab. And that's always been our goal here, to get to a CLIA lab. It's a lot of time and money, therefore. So we set out to do a multiplex testing here. And we set out with our first partner was Mesoscale Discovery for this MSD. So no booze here yet here with it. But we started with Mesoscale Discovery. We had the first cohort, we had about 300 patients, again, half cancer, half non-cancer there. We met with them. They gave us a prototype assay. They optimized it. It looked good. And that's when we got the 80% sensitivity, 81% specificity. They did another round of optimization, so now it's been optimized 2 rounds there, and they ended up giving this back to us. We tested in this cohort, and this is a cohort of about 300 patients again. This bottom cohort's interesting. It's the first cohort we did that was completely out of North America. So it was 2 cohorts from Japan, one from Nara, Japan, and one from Kyoto, Japan. And with this second round of optimization, we have a nice sensitivity, 85%, specificity, 81%. Then we met with them to say, okay, let's push this to the clinic, and we didn't feel they were the partners to take us to the clinic based on the timeline and the cost. And then that's when we reached out to Casey, and we were chatting with Blake as well from the Luminex, and Martin from the R&D Biotechny. And we set out on this course back in 2017 to flip from MSD to the Luminex platform. And that's what we ended up doing here. We met with R&D Systems. They gave us the prototype. They went through 2 rounds of optimization. And then in this study, we compared the optimized MSD platform with the optimized Luminex platform made by R&D Systems. And this is showing you head-to-head, the Luminex-based platform had the better area under the curve, 0.97, sensitivity 93%, specificity 95% compared to that of the MSD, 85% sensitivity, 80% specificity. So we felt like we made the right decision here. Plus, it wasn't going to take as long to get to the market, and that was our ultimate goal there. So at this time, again, submitting papers, submitting grants, we got comments back. You keep touting this as a liquid biopsy. Can you show me that what's in the urine is the same thing in the actual tissues? And that's what we did. So this study had about 300 patients. We had the tissue samples there, and we did immunohistochemical staining for our 10 biomarkers. We actually broke this into 2 papers. We scanned the slides with the Apereo system, and then we first analyzed it with the epithelial component, then later analyze it with the stromal component with some immune cells there. But this is one of the figures from the epithelial one that shows 9 of the 10 biomarkers were significantly elevated in the cancer compared to controls. In the stroma manuscript, all 10 biomarkers were significantly elevated in cancer versus control. So this is an actual representation of what's going on in the tumor. Then we started talking with our docs to see how can the clinical information we have help us here, because obviously the clinical information is quite valuable here. We actually started working with one of the experts in nomograms, Mike Catan at Cleveland Clinic, and he helped us construct this nomogram. And I'll just point you to this figure right here. So we took some clinical features here, age, gender, and race, and see how well that could help us predict cancer there. It didn't do that poorly there. Our molecular, the black one is the molecular, was better. The best was a combination of it. So now moving forward, we know let's incorporate some of the clinical features in it here that's relevant for our disease. So at this time, we had the assay optimized, again, thanks to working with Luminex and R&D systems. And then we did the analytical validation for it based on the FDA guidance paper, as well as the Q submission that we did with the FDA. So really, we want to know, is the assay doing what it's supposed to do? And sure enough, it is doing what it was supposed to do. It shows you everything that we had to check off there. Again, we've published everything there, so feel free to reach out to me for any and all of these publications. So now we've done analytical validation. Now let's look at early clinical validation here. So this is a study that we just did last year, about 350 patients presenting to my outpatient urology clinic for evaluation of bladder cancer. And this is the one that shows the sensitivity 93%, specificity 93%, negative predictive value 99%, positive predictive value 65%. So a few years ago, I asked the docs, what do you want to see in your biomarker there? What type of number? And they're like, I want to see high sensitivity, high specificity, high negative predictive value, high positive predictive value. I'm like, okay, that's going to be almost impossible to do there. What's your minimum that you need there? And they said high sensitivity and high negative predictive value. Sensitivity makes sense there. Why high negative predictive value, I asked? Well, they said it would be criminal if we missed a high-grade, high-stage, i.e. deadly cancer. We do not want to miss anything like that. So drive that negative predictive value up. So that's what we did. We have the algorithm that will drive this negative predictive value up, but it's going to be at the expense of the positive predictive value. So this is going to be a rule-out test here. So this is a rule-out test. This is a BCG setting. Again, I told you about this therapy that we use here. We kind of stumbled upon this. Some of my colleagues at MD Anderson was looking at their cytokine profile to see if it can predict response to BCG. Their cytokine profile didn't predict response, but they gave us access to their voided urine samples prior to BCG. And these are the numbers that we're looking at here. The sensitivity, 81%, specificity, 84%. So this, again, surprised us. We weren't thinking in this space here, and we didn't think it would come back something as good as this. This is what we ended up with this data and some of our other data here submitting to the FDA. We ended up getting breakthrough designation status for the FDA, and we just submitted this data to the EU early this year, and we have the CE mark for this in Europe here for predicting response to BCG. With that, we were able to get a few more resources, i.e. a better computational biologist, and he's been able to really refine this algorithm. And now we have a sensitivity about 90%, specificity 87%. This is just quickly showing that we tested all the assays with this ex vivo model here. So long story short, we took normal urine samples and we spiked it with normal cells. So this is normal cells, or we spiked it with cancer cells, or we spiked it with blood here. And the punchline here is this BTA stat assay. The BTA is supposed to stand for bladder tumor antigen assay. It started as an ELISA assay, then morphed to a point of care assay. It measures complement factor H. Complement factor H is in the blood. So what we notice here is that you can get a positive BTA test just by adding blood to normal urine there. So it wasn't anything cancer specific, it's a surrogate for blood. Similarly, NNP22, nuclear matrix protein 22. Why is there a nuclear matrix protein in the urine of bladder cancer patients? So long story short, if you had normal cells and you put it into the urines, you can get a positive test. Just like if you put cancer cells, you can get a positive NNP22. So it wasn't measuring anything specific for cancer, it was just measuring cellularity. Now, we would be remiss if we didn't throw the same rock at our assay here on Curia. So that's what we ended up doing here. For OnCuria, we did the same ex vivo model. The benign cells did not cause a positive test. The blood did not cause a positive test. individual biomarkers were increased, but it didn't flip to being a positive test. What did cause a positive test was having the cancer cells in it, but again, we're okay with that. That's what it's supposed to do, detect cancer cells within a voided urine sample. So now here at the end of the day, we have this one kit that can query 10 biomarkers. The only thing different is the algorithm associated with it, or the AI associated with it. So OnCure Detect, evaluating those patients with blood in the urine. So we have the 10 biomarkers, they're weighted differently, and we have age, gender, and race in that algorithm. OnCure Monitor recurrent disease. You have these 10 biomarkers, but you don't have age, gender, and race there, because that doesn't play a role in determining who's going to have a recurrent cancer. And then you have OnCure Predict, predicting response to BCG. Same 10 biomarkers, weighted differently, but now we have tumor grade and tumor stage in there because we know they play a role in predicting response to BCG. So one kit, different algorithms for the 3 different indications. So here we are, again, we want to make sure we do things in the clinic that makes it easy for the docs and for the pathologists there. So midstream voided urine sample, very common for us to get that in these patients there. The only thing that we ask differently is that this has to be stored at 4 degrees until we analyze it there. If it's not stored at 4 degrees, let's say it's out at room temperature for 2 hours, the proteins start to deteriorate at that time. But at 4 degrees, it can stay for a week. You can freeze it, and it stays for months there at -20. So obviously, the only thing we have to do before running it, gentle centrifugation, put it on the plate there. We only need 300 microliters there, 300 microliters, because unfortunately, we couldn't get all 10 biomarkers to play nicely within one well. So we actually have to have them spread out over 3 wells, or in this case, 3 panels there. And the output is a CSV file that we're all familiar with, right? This is the video here. Can you hit the video? So we ended up taking our algorithms and putting it in a web portal here. And so the web portal is now in the Amazon cloud, HIPAA compliant. And this video is showing that the CLIA technician here is uploading the CSV files into this web portal there. It's being analyzed in real time there. But before it's being analyzed, it does go through QA, QC to make sure the data is good going in. So it's really looking at the standards and controls and make sure that looks good. And then once that passes QA, QC, it pushes over to the algorithm. And you can see just in a short amount of time that I've been talking, it's ran this plate, and we ended up seeing the risk score for this one particular patient, 55%. So this is how we envision reading it out to the doctors. We're about to launch as an LDT literally just in another day or so here. Our LDT partner, CLIA partner, will be DiaCarta here. So with it, we have to read out to the, as the FDA wants us to do, dichotomous, cancer or no cancer, high probability of recurrence or low probability. So this is what we're reading out here, and then the risk score is there as well. So again, DiaCarta is about to launch this test. That's my Nonagen hat on. The clinical utility here. So we ended up doing a small study looking at 10 clinical vignettes here, and then showing the docs about using OnCure our test or using cytology here. And so the long story short with this one is shows that 31% of the docs said that they would reduce the number of other diagnostic tests ordered if the OnCure test was negative there. Largely, they were just omitting the cytology. On the flip side, if the OnCure results were positive, 27% of the docs said they would reduce the number of tests there. And again, it was mainly cytology not being run. So we thought this was interesting because this had not launched as an LDT yet. So the docs haven't been able to order it and use it to treat their patient. But it shows you how ripe the market is to get a more robust test in here to help care for our patients. And the last thing to talk about on this slide is the risk score. The docs liked the risk score because they said the risk score made the test more actionable. Well, we like to hear that it's more actionable, but the insurance companies are the ones that's really going to want to hear that they want to pay for a test that's actionable. We have several clinical trials ongoing. So the lab's funded with an R01 grant and a UH3 grant and a U54 grant. So we have several clinical trials ongoing for our indications that I chatted about. And this is showing you how we stack up against our competitors. So we're head and shoulders above our competitor with sensitivity, specificity. We're the only ones in the game for predict. No one else is looking at predict. And the docs like that because it kind of differentiates us. And again, we're not going to rest on our laurels to be an LDT. We want to go for the IVD, so we want to do the FDA clearance and approval here for our indication because it's going to have a broader uptake, not only in guidelines, but around the country and around the world. So, in summary, OnCure addresses an unmet need by identifying patients who will not respond to BCG. Again, if you're not going to respond to BCG, it's futile to keep throwing this drug at you. And again, we have a shortage of this drug. Let's pivot this patient to a therapy that's going to actually work for them, and OnCure can help with this. OnCure has an unprecedented accuracy, which will give urologists a higher level of certainty compared to existing urine-based diagnostics, i.e. urinary cytology. And then lastly, with multiple applications under investigation, OnCure has the potential to cause a major shift, not only the way we manage bladder cancer here in the United States, but the way we manage bladder cancer globally. So what's the saying? It takes a village. It definitely did take a village because I didn't do any of this stuff here. I directed people here. But over the last 10 years, we collaborated with key people here, not only in the US, but globally here. And I definitely want to point out that you can't see it so much on the bottom, the 8,000 patients that ended up providing samples for all of our testing and all the grant funding mechanism there as well. Thank you very much for your time and attention. I look forward to answering any questions you may have.